Hi, everybody, and welcome to today's Seed World Innovation Series webinar. My name is Mark Zinkowitz, and I serve as Associate Editor for Seed World. And today I'm happy to be your host. Today's theme is Advanced Machine Learning for Predictive Plant Breeding. I'd like to take a minute to thank Computomics for partnering with us on this Innovation Series webinar. We have a great lineup of speakers for you this morning. Presenting the webinar today are Ruth Mays and Christian Dreischer. Ruth is Director of Global Business Strategy and joined Computomics nearly 3 years ago and manages all aspects of the business and commercial strategy. She has a scientific background with a broad knowledge of applied research in genomics and the life science industry, and experience in organizational leadership, building and managing teams across multiple sectors. Christian is scientific project manager at Computomics. He studied bioinformatics and wrote his research thesis at the Max Planck Institute for Developmental Biology. His experience in genomic data analysis using machine learning techniques together with his project management skills, led him to advance the technology on accurate phenotype prediction and accelerate plant breeding for a changing climate. Now, during the presentation, you'll likely have some questions for our two speakers. Please type these into the chat box at any time, and we'll address them during the Q&A session we'll hold after the presentation. Also, we'll have a few poll questions for you throughout the presentation. These will pop up in your chat box. And today's webinar is being recorded and will be made available at seedworld.com. Following the proceedings, we'll also send you an email with a link to the recorded version of the webinar. Now, more than ever, data is enabling predictive plant breeding. Today, you're going to learn why it's important to build a robust data set in plant breeding, what a cost-effective training population looks like, what the advantages are of machine learning technologies over classical statistical methods, how you can unleash the accuracy of machine learning-based prediction methods in your breeding, and what questions machine learning can solve in hybrid line and clonal breeding systems. Ruth and Christian, good morning. Thank you for joining us today. The floor is yours.
Thanks, Mark, for the introduction. Computomics was founded out of the Max Planck Institute of Developmental Biology to bring new and innovative tools to industry. We have completed way over 135 projects across 12 countries, and we are disrupting plant breeding with our machine learning technology. I'm Ruth Mays, Director of Global Business Strategy at Computomics. The webinar today would like to showcase the advantages that machine learning technology provides over traditional statistical methods. We will also explain the difference and benefits between machine learning and genomic selection. The Computomics team consists of bioinformaticians and machine learning experts who have spent many years developing and applying machine learning to breeding to develop our Exceed Score technology. I'll start the webinar by showing how we support clients to build a robust data repository and how we can build a strategy which will enable breeders to access all of the genetics within their breeding programs. The technology is designed to identify and progress elite genetic lines through a breeding pipeline and to increase the probability of accelerating elite candidates into a commercial pipeline. I'll then hand over to Christian, who will provide a comprehensive technical overview of the technology and explain the benefits of machine learning over other traditional statistical methods. Chris will also show how the technology can be applied for hybrid, line, and clonal breeding programs. We'll then finish off with a summary and a Q&A session. Our aim is to allow every breeder to access all of the genetic potential within their breeding programs. We work with breeders to build a genotyping and phenotyping strategy which results in a robust data repository which accesses all of the genetics and future-proofs their business. We evaluate any current phenotype data and also any genotype data available, and we then use this information to build the data sets that enable genomic predictive breeding. We advise on the amount and type of data required, and we also offer the best advice on which genotyping technology fits your program. The aim is to advise on generating cost-effective data sets which include the amount of phenotype data required and all of the measurements required. These can include the number of times the measurement needs to be calculated and the time duration between each measurement. This will optimize your phenotype strategy and reduce your overall phenotype costs. The next step is to then create populations to enable genomic predictive breeding. In the next couple of slides, I will show examples on how to enable both line breeding and hybrid breeding. However, machine learning can be tailored for all types of breeding structures. Before we move on to the next slide, we'll have a quick poll question. An ideal training population includes a broad set of information, which also includes mediocre lines, as the technology needs to understand performance across the whole spectrum of the breeding program. The model independently learns from the dataset and makes distinctions to make accurate predictions. The criteria must include genetic diversity, the same performance distribution in both training and target data sets, and the training lines need to be genetically related amongst each other and to the targets. They can also include parents of crosses, sister, parent, and grandparent lines, and previous generation crosses. Phenotype measurements need to be measured under similar conditions with the same locations and same years. We can help to optimize population size, and generally we ask for as much genotype and correlating phenotype data as possible, as this will help to train a robust model. But we can make accurate predictions from just hundreds of correlating lines. For line breeding programs, to enable genomic predictive breeding using machine learning, you need to build both a training and target population data set. The training population should be representative of the target data set. The training set includes genotype and phenotype information based on previous years. The target set reflects predictions for forthcoming years. The data sets can also include additional data, such as climate, temperature, wind speed, location, soil analysis, and agronomic practices. Additional data sets can offer location and climate specific predictions and increase the accuracy of the model. You need to include representative lines that you genotype and phenotype so that the model can generalize and predict all of the variants within the population. A robust marker set is required that represents the desired traits of interest. The data set needs to have good marker representation that needs to interpret the trait. This is especially significant for complex polygenic traits like yield, as yield is governed by dozens of genes. We can also identify trait correlations and use these to improve the predictions. The model is very adaptable and scalable with regard to the number of markers and the population size. The model can also be adapted for all breeding programs, including clone breeding. For hybrid breeding, a robust marker set is required that represents the desired traits of interest. We can work with the breeder to create both genotype and phenotype datasets. We then create a double haploid setup, created with homozygous parents. The machine learning model can be used to identify elite testers with the best general combinability, focused on accessing the best genetics within your program. Both male and female testers can be optimized simultaneously to create a training population. Christian will go into this in more detail further on in the webinar. Once the model is established, you can use offspring predictions with both general combinability and specific combinability as new parents for the next cycle. Each cycle results in genetic gain as you are advancing hybrids from previous cycles that become parents, advancing both GCA and SCA each cycle. The model can be adapted for all hybrid setups, including segregating populations, and for more complex crops and programs that include restorer lines, such as rice and wheat. This slide provides a visual overview of how we start to work with breeders to create the most suitable data sets. On the left, the breeders supply their knowledge, expertise, and any existing data. The Computomics team identify which lines to genotype and the best genotyping technology, and also on creating the most cost-effective phenotyping models, which enable consistent, accurate predictions each cycle. The consultancy program enables access to the machine learning exceed score technology and can provide a fast track to implementing genomic predictive breeding. We work with the breeders to understand what they want to achieve cycle upon cycle. With robust data sets of both genotyping and correlating phenotyping information, we work with breeders to help them to optimize their breeding goals. With the right breeding structure and data, we can ensure that breeders are identifying all of the elite genetics within their breeding programs. Ultimately, breeders will be increasing their probability of success and escalating many more candidates into the commercial pipeline. We are actively pursuing with real breeders entry strategies into using breeding methods, fine-tuning population structures and genetic makeup to optimize the analytical capabilities of the technology. I'll now hand over to Christian, who will explain the technical aspects of the technology and also the differences between statistical methods and machine learning.
And we'll just wait a moment for Christian. Again, if you have a question, please type it into the chat box and we will answer your question at the very end of the presentation.
So, thank you, Mark. Thank you, Ruth, for this overview. I will focus more on the technical details and give you examples of hybrid and line breeding programs that we supported. So, why do we use machine learning for genomic prediction? We were approached by breeders who weren't satisfied with the results and insights they got from classical statistical models like RR-blob, G-blob, or other flavors. The environment can have a major effect on the phenotype, and classical statistical models have a hard time accounting for this. So the environment can have a major effect on the phenotype, and classical statistical models have a hard time accounting for this. The same is true for complex traits. And as a company with a foundation in machine learning, we started to develop a machine learning-based technology that would address these shortcomings of classical statistical models. So what are the benefits of using machine learning in genomic prediction? The prediction accuracy for monogenic traits is pretty similar between classical statistical models and machine learning approaches. But machine learning approaches can considerably improve the prediction accuracy for complex polygenic traits, such as, for example, yield or oil content. In contrast to classical statistical models that use a simplified kinship matrix, machine learning approaches model the influence of each individual marker and of marker combinations on the phenotype. By doing so, they are able to model non-additive effects, such as epistasis or dominance. And we also get a better understanding of GCA and can identify cases of SCA. Machine learning technologies can integrate very heterogeneous data, and so we can incorporate environmental measurements such as precipitation, temperature, soil type, etc. As machine learning approaches determine the importance of each factor that goes into the model and of the interaction between these factors, we can also tell which environmental factors influence the phenotype, meaning we can model G by E. Machine learning technologies can optimize for multiple traits at once and make use of trait correlations to improve the prediction accuracy. So, in sum, machine learning technologies are a better approximation of the biological reality than classical statistical models. That's why we've developed the machine learning-based technology XSeedScore to help breeders reaching their goals more efficiently. XSeedScore needs no a priori knowledge about the population structure. We can use this information, but it's not a prerequisite to make good predictions. We integrate environmental data in our prediction models from public services like NOAA or provided by the breeder. As I mentioned before, we can model the influence of all factors that go into the prediction model and can tell you which environmental factors and markers influence a trait the most. And we're able to predict for untested locations or tell you which lines will fare well under which environmental conditions. By using allele dosage, we can also work with polyploid organisms. Breeding programs are vastly different, and the data we get reflects that. Difference in population size, in the number of phenotyped and genotyped individuals, in the number of markers, or with regards to the genetics. We don't have a single one-fits-all solution, but apply different machine learning techniques to address these different setups, and are therefore very flexible with regards to different input data. One of the core functionalities of XSeedScore is the simulation of millions to billions of offspring to assess the combining ability and the performance of parental lines. As Ruth mentioned before, a good training population that reflects the genetic and phenotypic diversity of the breeding program is really key for good predictions. Machine learning approaches learn from data, and with increased data quality and quantity, the prediction accuracy improves. The diagram on the right-hand side shows results from a breeding program we're supporting for several years now. By adding data from previous years, that is historical data, our predictions gained in accuracy with each year. We use statistical models as a baseline for prediction accuracy and are always developing this technology further. So now I would like to give you some examples on how we support hybrid breeding programs. Which questions do we solve in a hybrid breeding program? We support you in identifying the best testers. We can tell you which parental crosses will result in superior progeny. And we give you a good understanding of GCA and can identify cases of good special combining ability. One question that comes up in the beginning of a hybrid breeding program is the right choice of a tester or testers. This diagram shows an example of a conventional hybrid breeding program. We have female heterotic pools AB and EF and male heterotic pools CD and GH. Now we pick a set of testers from each side and create all possible hybrids with the other side. Based on field tests, new testers are selected and new crosses are made and evaluated in the following years. This has to be done multiple times to assure that we did not pick the wrong tester and get good combiners from both pools. In this case, the whole process took about 8 years from start to finish. A breeder can test only a small number of test hybrid combinations, as the field space is of course limited. With machine learning, we can create a model based on the set of all hybrids and testers and simulate and predict the phenotype of all possible combinations between them. This way, we get a good sense of the combining ability, identify optimal testers, and simultaneously improve both sides of the pedigree at once in just 1 to 2 cycles. We can also identify low-performing lines early on, so that they are not planted in the field in the first place. In the breeding scheme of one of our customers, this saved half the time of the breeding pipeline and increased the number of candidates 10 times. The classical approach would only evaluate the performance of top cross hybrids, as there is limited field space. As we simulate all possible crosses, our search space to identify high-performing hybrids is vastly larger. We can simulate and predict millions to billions of genotypes within a couple of hours. This is much more challenging with classical statistical models, as the creation of a kinship matrix of such a vast number of individuals is computationally extremely demanding and takes a very long time. As field space is limited, in a classical approach, you would only cross top-performing parents, field test them, and identify a couple of candidates to take into the next cycle. The hybrids of these crosses are represented by the dark gray cells in the left-hand matrix. And the red ones are the top-performing ones, as predicted by, for example, classical statistical models. The matrix on the right-hand side shows the exceed score approach. As we simulate the crosses between all parents, we dramatically increase the possibility to identify high-performing hybrids, and we can also identify cases of good special combining ability. The red crosses symbolize the top 10% of top-performing lines. And all the red squares outside of the upper left box are cases of good special combining ability. So in this example from one of our customers, we had 600 males, 150 females, and 1,000 hybrids as a training set. The data came from several years and locations. We simulated and predicted the phenotypes of 100,000 virtual hybrids, a couple of million, including the different locations and years. The heat map shows a subset of this data. You see the predicted performance of all parental combinations ranked from high performing in red to low performing in blue. The parents were clustered by the hybrid's performance, and so it's very easy to spot parents with similar performance. And it's also very easy to calculate the general combining ability by just summing of the rows and columns. It's also easy to identify cases of good special combining ability. Based on the clustering, we can identify the best line from each cluster and avoid taking redundant lines. So, in summary, by predicting not only the performance of top-cross hybrids, but of millions of virtual hybrids, we get a much better understanding of DCA and can identify cases of special combining ability. This way, we can identify superior parents. And as I've highlighted in the beginning, our machine learning models can take non-additive genetic effects into account and therefore provide superior model accuracy. We have a very successful collaboration with Beck's superior hybrids for many years now, and they allowed us to show real numbers from one of their breeding programs we worked on together. The top row depicts an average maize breeding program. The bottom shows the maize breeding program using the before-mentioned approach, where we advance both sides of the pedigree at once by simulating all versus all double haploid crosses. Our training population for exceed score was 2 years of field data. From that, we built a machine learning model. In contrast to performing field tests like in a classical approach, where 2,000 lines are picked for field testing, we simulated 30,000 virtual hybrids and basically skipped the first year of field trials. From these 30,000 virtual hybrids, 400 were selected and planted based on good performance prediction. An incredible 52% of those were advanced into the third year as they showed good performance on the field. And within just 2 years, 33 lines went into pre-commercial tests, 6 into commercial evaluation, and 1 was launched commercially. This was only possible by advancing both sides at once and by doing large-scale hybrid simulations. The key success factor for BECS is that they dramatically increased the possibility to identify a new elite line. Remember, we start with a pool of 30,000 hybrids to choose from, compared to just 2,000 to find a new superior variety. So, with this example of a very successful collaboration, I will end the hybrid breeding part and will go on to the line breeding part. So in a line breeding program, we want, for example, to know which parental combinations will lead to offspring with favorable trait combinations, such as high yield and early flowering. Can we identify heterotic groups in our population? And which crosses and how many offspring per cross do we need to make to be more efficient in identifying successful crosses? Every breeder is limited by field space and can test only a couple of parental crosses. So as we do in a hybrid breeding program, we perform large-scale offspring simulation. In this toy example, we'll start with a 50 by 50 cross and have simulated over 1.25 million offspring in the 4th generation. By looking at the performance of thousands of offspring per cross, we get a very good assessment of the cross's potential to produce superior offspring. We can work with only the parental genotypes and phenotypes, but as I mentioned earlier, machine learning models really benefit from data quality and quantity. And by phenotyping and genotyping selected offspring, or by including historical data from genetically closely related lines, we can increase the prediction accuracy. So within each generation, we sort the offspring by performance. By looking at thousands of offspring per cross, we can tell you early on which of the lines you can discard, as their offspring will perform poorly, and which of the crosses to keep. Exceed score tells you the most important markers influencing a trait. So we can identify beneficial haplotypes, that is, marker or allele combinations that are associated with, for example, high yield. This knowledge can be used to screen your germplasm or could be used as gene editing targets. So as mentioned before, we can include weather data to improve the prediction accuracy and also to predict for untested locations or to tell you which lines will fare well under which environmental conditions. In this example of 10 locations, the number of simulated offspring grows to over 12 million. This is computationally very demanding for a classical statistical approach. So here we have another example from one of our customers, where we optimized for 2 traits at once. The breeder was interested in plants with high yield and early flowering. Yield and days to maturity are usually highly correlated. This is what the plot on the right-hand side shows. Each color cloud basically represents the simulated offspring of one cross. We could identify crosses like that in the top left corner for that yield and flowering time were basically uncorrelated and showed high yield and early flowering. We can optimize for any trait combination by also keeping genetic diversity in mind. This is assumed in view of the previous plot and shows only the simulated offspring of one cross. We could identify 2 heterotic groups within the offspring, and by looking at the marker influence on the traits, we identified 3 SNPs that distinguished between these groups. The breeder later confirmed these SNPs via a QTL analysis. This again shows the power of the machine learning approach. This is another example from the initial color cloud, a cross between a parent with low yield and low maturity and one with high yield and high maturity. As you can see, there are quite a few lines that outperform their parents. And by looking at the relation between low and high performing offspring, we can estimate how many offspring you need to generate to have a high chance of producing a new elite line. So clonal breeding is basically a special case of line breeding. The goal is the same. We want to identify high-performing crosses and discard low-performing early on. The main difference is the genetics. In a typical line breeding program, you're trying to get mostly homozygous lines in an F4, F5, F6 generation. In clonal breeding, the plants get propagated clonally after the first generation and don't change their genetics. The plants are often massively heterozygous and in some cases also polyploid. That means that we have to simulate a vast amount of offspring in the first generation, tens of thousands of offspring per cross, to assess the parent's performance to produce superior offspring. Doing this allows us to identify good and poor performing crosses and to identify parents with good combining ability that might be used as parents in the next cycle. So with this, I close the technical overview. Our main goal is to support you reaching your breeding goals more efficiently. That's why it is important to us that our results are easily understandable, interpretable, and can be used by you right away. That's why we tailor our reports, output formats, and visualization to your needs. We offer a plethora of ways to explore the data visually in interactive plots that makes it easy to extract the information you need and to make informed decisions. With that said, I will close the technical overview and hand over again to Ruth, who will sum up this webinar and lead over to the Q&A session.
Thanks, Chris, for the technical overview. And so, to summarize, with Exceed Score, you predict non-additive effects, including modeling epistasis. You also have the ability to predict both general combinability and specific combinability. As we look at the individual marker importance of the trait, this provides the knowledge to make accurate predictions. Multiple data sources can be incorporated into the model. These can include anything from soil analysis, wind speed, and temperature, and even farming practices. The technology can interpret data and accurately predict for specific locations and environments, including untested locations, which makes it very effective for future climate change. The technology also understands trait correlations and makes improvements by identifying genes that impact specific traits. Large-scale simulations mean that we simulate millions of possible crosses and can accurately predict candidates to escalate through the breeding pipeline. The model is very adaptable and scalable with regard to the number of markers and the population size. You can increase the probability of success by creating population sizes that can provide the best prediction accuracy and also escalate more candidates with elite genetics into the commercial pipeline. Thank you very much all for your time, and we'll now have the Q&A session.
Thank you so much, Ruth and Christian, for the great presentation. And thank you to our audience for joining us today. If you have a question, please type it into the chat box. First question, in general, what is the prediction accuracy for your untested locations?
Yeah, that's a good question, and this always depends on the breeding program, the trait of interest, and the data we have. So with very good data, we could reach prediction accuracies up to 0.85. But for machine learning, it always is the matter of good training population, of good data, especially the phenotyping data must be of good quality, and the environmental measurements should be sufficient.
In line development, how can we select offspring selection with the help of machine learning?
Well, as I showed, we do these large-scale offspring predictions. So for each cross, we have thousands of or tens of thousands of offspring that we advance to a F4, F5, F6 generation. And we do this as biologically sound as possible. So if possible, we use, for example, the recombination frequency to simulate the offspring as biologically realistic as possible. And by looking at the performance of thousands of offspring of a cross, we can determine the parent's performance or the parent's probability to produce superior offspring.
What is the size of the data that you generally need in order to do this?
Well, this also depends on the breeding pipeline, on the population. We have worked with very little data, so with just a couple of 100 markers, very successfully, but those markers were handpicked beforehand by QTL analysis. So in a hybrid breeding program, we can work with like 1,000 lines and with just a few hundred markers. And we've seen a peak in prediction accuracy when we reach 10,000 markers or more. Then the prediction accuracy didn't improve. But we've also worked with really large sets of tens of thousands of lines and really large marker sets.
Since machine learning requires large amounts of data, who is collecting the data to make up the system?
So as Ruth mentioned before, we can assist you in generating the right kind of data, so defining a training population, supporting you in choosing the right kind of genotyping technology. We also can subcontract the genotyping for you. The phenotyping data, that's a very crucial part, must come from the breeder, of course. We can't take over that part. Environmental data we can retrieve from public sources, from NOAA, for example, from satellite data. But if the breeder has local weather stations, that's of course preferred.
What is needed as a precursor to utilize machine learning to place inbreds into heterotic groups?
Well, that's also a good question. The heterotic groups are usually defined by the breeder. But we can, by looking at these all versus all simulations, give you an assessment of your lines, and can also tell you in which heterotic group a new line might belong.
What would be the best strategy in getting good predictions? My breeding program has only a small number of parental genotypes to work with. Can you offer us some insight there?
So in the beginning, when we work with a new breeding program, we do a thorough assessment of the genotypic and phenotypic information. So we look at the genetic diversity, we look at the phenotypic distribution, and we make some test runs to see how the prediction accuracy for the traits of interest is. And based on this assessment, we can tell you if the data is already sufficient for your traits of interest, or we can support you in generating or advising you on generating better data. So can tell you if you need better genetic diversity, if you need more markers, for example, or also which of the environmental measurements you need to make to support the prediction.
What specific type of genotyping is best for your process?
We are independent of the genotyping technology. So we've worked with GBS data, we've worked with chip data. There are many flavors of producing genotyping data, and we've seen a variety, and can work with every kind of genotyping data.
How many years of phenotypic data and from how many locations is ideal in order for it to be subject to prediction?
So if the data you have is genetically related, each additional data point helps. So if the historical data we can include in the model is genetically related to your current population, then every additional data point really helps. If the data is from unrelated lines, so from unrelated genetics, then additional data doesn't really help. But as I've shown in the beginning, we see gain in prediction accuracy by adding historical data from previous years. And the prediction accuracy increased with every year.
Can you talk a little bit about the machine learning algorithms that you've implemented in your work?
Yeah, we use different flavors. As I said, different breeding setups require different technologies or different approaches. And so we use different kernel-based technologies. We also have implemented different deep learning algorithms. But yeah, I can't go into the specifics of these algorithms. That's our IP. But yeah, we use really a broad range of technologies. We compare them to each other and are constantly developing them further.
For a given crop like corn, how many SNP markers do you include in your genotype characterization?
So could you repeat that? The audio was scrambled up.
Sure. For a given crop like corn, how many SNP markers do you include in your genotype characterization?
Okay. Yeah, so for a current breeding program we have supported now for many years, we have started with 5,000 markers, was a 5K chip, and this has now bumped up to a 30K chip. And we see an increase in prediction accuracy. So it's not dramatic. But yeah, so that's the range we've worked with in maize. And we've also, so for this transition, we've took the 5K chip and imputed the genotypes up to this 30K chip. So that's also something we can do.
Do these predictions work in the case of multi-parental crosses, or does it require only biparental crosses?
No, so as we don't compute this kinship matrix, but we really look at the individual marker effects, we can also work with multi-parental crosses.
How much better is machine learning over statistical models, and can you provide an example?
Yeah, so for monogenic traits, for simple traits, we don't see a real difference between classical statistical models and machine learning approaches. I think the real benefit of machine learning approaches is the modeling of the individual market influence and of the interaction between different markers. So modeling the effect of 2, 3, or 4 markers at once on a trait. So the real key benefit is predicting for complex polygenic traits, yield, oil content, maturity. And there we've seen prediction increase in 0.2, 0.3, also depending on the input data we get.
If a breeder has a particular rate of genetic gain in percentage points, can you frame your support around this goal? If so, what happens if these predicted gains are not realized?
So we work very closely with the breeder and define goals together and make a realistic assessment of the data he has and the goals he can reach with that data. So if we think from our experience that it's possible to reach this goal, we are confident that we can do this together. But we have a really good assessment of what's possible with a given data set now.
Every year, yield performance might be due to different gene networks. How do you take this into account?
So with each breeding cycle, so if you add new material, new genetic material, we train a new model. And each model will, if the genetics differ, each model will look differently. So the markers will be weighed based on the data it gets. And if we, for example, include weather information, and we see that there was maybe low temperature in the beginning of the breeding cycle or more rainfall, then machine learning models can pick up the signal and identify new marker combinations that influence this trade. So new data will require a new model that will look differently from the previous model if the biological reality changed.
How many years, along with locations and morphological data, is required for prediction?
We start with 1 year of data. I think that's very similar to the previous questions. The prediction accuracy just increases with the amount of data. So we can work with the data from just 1 year. And by that, by adding historical data, the prediction accuracy will improve.
In a commercial breeding setup, how long would it take to realize the benefits of this?
Probably depends on if you're in a hybrid or line breeding program. I think in a line breeding program, it takes more time. Breeders we've worked with see benefits, I think, after the second year, if they can, when we identified, for example, low performing crosses that they didn't have to plant in the field, or could identify really beneficial crosses that they would then plant instead.
Now, what if you have a situation where there exists there's a complex but unknown population structure, and the environment of the examples is poorly controlled or changing from time to time? What would you do in a situation like that?
So we don't need the population structure information, as we don't rely on the kinship matrix. But we look at the genetics of each individual, and within each individual we look at the marker influence. So population structure is not required. The genetics should be related to what you want to predict. And it really comes down to a good training population. The population structure is often unknown to us, and we don't need this kind of information. as we really learn from the individual market effects.
Yield is obviously an important target, but also fruit quality attributes. How do you estimate multi-trait selection or index selection?
Yeah, we can optimize for multiple traits at once. So if you have 2 or 3 traits that you want to improve, we look at, for example, trait correlation. And there are machine learning technologies or techniques that optimize for different goals, in this case, different traits. And it's independent of the kind of trait. So we can optimize for fruit quality and, for example, disease resistance at once.
Do you offer a pipeline for open pollinated varieties?
Yes. So we, for example, work with soy breeders, and the technology is independent of the crop.
What percentage of heritability or percentage of PVE do you consider good for selection?
I mean, the prediction gets easier the more heritable a trait is. I'm not sure what the goal of this question is. So I think
how much percentage of heritability PVE do you consider good for selection?
I think that's mostly up to the breeder, really. The prediction accuracy increases with the heritability of a trait, so we can get better prediction accuracy the higher the heritability. But the choice really has to be made by the breeder.
How do you deal with epigenetic influence in prediction? How do you take it into account?
So we've worked with epigenetic markers for a long time and have developed our own technology to identify epigenetic markers and epigenetic difference, so methylation differences between different organisms. And we've already tried to incorporate methylation markers into genomic prediction models and could see an improvement in prediction accuracy. So methylation markers are, as other environmental measurements or other metadata, just another feature that goes into the machine learning model, and that will and so the machine learning model evaluates or determines the influence of these markers as it does for genetic or environmental factors.
I have a question about cost efficiency. In general, how much more cost efficient is this compared to using statistical models?
Well, depends on our success, but that's a question for the breeders we've worked with, and that's something where we don't have great insight to. We rely on their willingness and eagerness to work with us each year anew, and we've worked with several breeders for several years now. So it must have some benefit to them, but I can't, tell you the monetary value it has.
Yeah, we can't exactly give you a cost implication, but obviously you're streamlining your breeding program. you're optimizing your testers, so you're saving time. So that obviously is a cost-effective factor, and obviously you're using your nursery and your field plots much more intelligently because you're highlighting your high genetics. So that obviously saves you a lot of time and a lot of money as well, because you're discarding the low performers very quickly.
Have you been able to tackle genomic selection for vegetable breeding specifically?
We've also worked with vegetables. Yes, sometimes they're a bit more challenging with the traits. The traits might be more complex, but as I said, we're basically independent of the crop. We look at the genetics. We look at the marker influence on a trait. And so if it's a wheat or a tomato or pepper, this is basically the same for us.
I have a question regarding breeding in the hemp or cannabis space. Have you guys worked on anything in that area?
Yeah, we're working with a breeder who went into cannabis breeding just recently, and we're collaborating in building a good breeding pipeline, starting from a good starting population, identifying good markers for the traits they're interested in, and then really scaling this up to a big breeding program.
Yeah, again, cannabis is a very complex crop, but we can still optimize traits. So as Chris already said, it's irrelevant of the crop. The technology can be tailored for pretty much any crop.
And I'll just see if we can tackle maybe one more question before we sign off for the day. We just hit the 1 hour mark. So thank you everybody for joining us today. It's been a fantastic webinar. And if you have any additional questions that we're not able to get to, please feel free to contact our speakers. Their contact information is on the screen as we speak, and they would be happy to answer them for you. Let's see. How efficient is prediction breeding for virus resistance in vegetable crops?
Resistance is usually a more simple trait. We have many known disease-resistant genes, and if they are known, we can fix this in our model. So we can say that a specific marker that stands for specific disease resistance must be there in our selection. So we can optimize for disease resistance by simultaneously select for other traits of interest. So we can basically keep this as a fixed factor.
Thanks for that, Christian. Well, that's all the time we have today. I'd like to thank Ruth and Christian for joining us today, and also a big thank you to Computomics for making this webinar possible. I'd also like to thank you, our audience, for participating today. We hope you found this information valuable. Again, a recording of the webinar will be available very soon on seedworld.com. Thanks again, and I hope you have a terrific day. This is Mark Zinkowitz of SeedWorld signing off. Thanks so much, everyone.
Thank you very much for listening. Have a good
Thanks,
day.
Ruth.
Thank you.
Yeah. Bye.
Bye.
Bye for now.